Okay, so again, folks, just for the movie, this is Thursday, October 17th. This is lecture, and we're doing Microbial Genetics translation, and we think we can start Microbial Genetics Part 2 today, and there are 2 new handouts which will be passed out. Okay, so folks, you will recall we are talking about gene expression, and what are the 2 stages of gene expression that we described? Transcription, right, where DNA acts as a template to make RNA, right, and we're going to be specific and say, at this point, we're going to make mRNA, because mRNA acts as a template for which process? Translation, right? So you guys, we had just started translation, and we said translation is really, it's really cool, but it's really complicated. So our approach is to use translation as an onion, right? We're gonna start simply with the outer layers, and then we'll start peeling the onion back layer by layer to get more and more detail. Okay, so let me advance here to, so you guys, what is the key to translation? What permits the cell to use a mRNA base sequence and then translate the mRNA base sequence into amino acids? What's the key to that cool event? The genetic code. Awesome. Awesome, you guys. So the genetic code, the one-to-one relationship between codons, and codons are triplets of nitrogenous bases in mRNA and a specific amino acid. And as we had briefly mentioned, you guys, there are 3 triplets that won't code for an amino acid, so we'll come back and talk about start and stop codons. But first, again, you guys, we just want to do our basic review. So we said that on lecture exam 2, you'll have an mRNA sequence, and I'll ask you to translate it into an amino acid sequence. So the genetic code table will be on the exam, right? But we have to know how to use it. That's the important thing. So you guys, so let's just review that really quickly. Let's say this is our mRNA, and let's say this is the 5' end and the 3' end. And again, folks, initially we won't talk about start and stop codons, right? We're just going to do this really simply. Okay, so folks, remember that codons are triplets of nitrogenous bases. So this is a codon, and we always number the codon starting at the 5' end. So this is codon 1, codon 2, codon 3, codon 4. Right? And we said within each of the codons that each of the nitrogenous bases has a position. So 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. Right? So what we want to do is ask ourselves, when the cell is translating this mRNA into a little polypeptide of protein, what will be the amino acid sequence, right? So to do that, we're going to use our mRNA codons and our genetic code to see how each of these codons will be translated. So this arrow means translation, right? How each of the codons will be translated into a specific amino acid. So you guys, and again, we'll pretend we aren't talking about start and stop codons yet. So you guys, do you remember how you would know which amino acid the codon AUG, which amino acid it would be translated into? Okay, so remember we have positions AUG, so we look at the genetic code table. So this is position 1, so we find A, so we know it's somewhere in this row. Position 2 is up here, so AU, so we know it's here at the intersection. And if we wanted to, we could go over here to find the 3rd position, or we could just do it by inspection. So AUG would be translated into, MET stands for methionine. And again, folks, on our lecture exam 2, your genetic code table will have the full name of the amino acids. You don't have to memorize the abbreviations, okay? So AUG, you guys, in the ribosome would be translated into which amino acid? Methionine. Yeah, good. We'll just say methionine for right now. Good. All right, you guys. And now just take a few seconds to yourself. Which amino acid will the codon CGC be translated into? So ARG is arginine. Okay, so that would be the second amino acid. And we're going to throw in a little bit more here, you guys. This is occurring in the ribosome, right? And within the ribosome, that ribosomal RNA, that ribozyme, is going to catalyze formation of what kind of bond between these two amino acids? A peptide bond, right? And what's really cool, again, we just want to remember, you guys, it's ribosomal RNA that's catalyzing formation of the peptide bond. So it's a ribozyme. Yeah, very cool. All right, folks, so what about the codon UUU? Which amino acid will UUU be translated into? So here you go, UUU. Phe is phenylalanine. Good. And again, folks, the ribozyme will trigger or catalyze formation of what? Yeah, good, all right. So folks, we can see now that we're getting an amino acid sequence, and the way the mRNA codons are read, right, that will represent the order of the amino acids. So this would be, for example, the N-terminal end, the start of our amino acid, excuse me, the start of our protein, and then this would represent the carboxy terminal end. And remember, you guys, it's the order, the sequence of amino acids that's so crucial to determine the functional conformation, right? So the order of the codons is really important. The order of the amino acids are really important. Okay. Now, folks, what about UAG? What about UAG? It's a stop codon. What do you think that means, a stop codon? It's going to stop translation, right? We are going to make it really simple, you guys. We're going to say that when the ribosome hits a stop codon, we're going to say the ribosome disassembles, it releases the mRNA, and it releases the newly formed protein so it can fold into its functional conformation. This was just an example, you guys, right? Okay. But with that introduction, you guys, of a stop codon, let's back up and look at the previous slide that had a little bit of like summary about the genetic code. Oops, went backwards. Okay, all right. So you guys, if I ask you what's a codon, what are you going to tell me? Right, it's a triplet of nucleotides or a triplet of nitrogenous bases, right? And we're talking about translation, you guys. So there's going to be a codon that is the start codon. It tells the ribosome where to start translating the mRNA into a protein. Now, just to give you a little bit of bigger picture here, you guys, so let's pretend this is our messenger RNA just produced by the ribosomal RNA, right? 5' to 3', yeah, okay? And an important sequence near the 5' end is going to be called the RBS. Had we talked about this already, folks? I apologize. The RBS, okay. RBS stands for ribosomal binding site. Right? So the ribosome is scanning messenger RNA, looking for this ribosomal binding site. It binds, and then, folks, it starts searching. After it binds, it starts searching for the first AUG. Right? So the first AUG, that's called the start codon, and that's where the ribosome will start translating all the remaining codons into amino acids. So it doesn't start right where it binds, and then it starts scanning, and as soon as it hits the first AUG, it'll start translation. Okay, so the first AUG is called the start codon, and then, folks, just as we said, if we have a start codon, we have to have stop signals. And what are the 3 codons that are the stop signals telling the ribosomes to stop? UAG, good. UAG, and what else? You don't have to memorize these, folks, because it'll be on the genetic code table. Will be, yeah. So what are the 3 stop codons, you guys? UAA, UAG, and UGA. And again, we're going to make it simple. Just say when the ribosome hits a stop codon, it disassembles, releases the mRNA, releases the newly formed protein, right, and translation is finished. Good. Okay. And again, folks, we'll toggle back and forth here. All right, and then key to translation, as we said, was the genetic code. All right, now, folks, there's a little bit more detail here. So in prokaryotes, so for example, in bacteria, the start codon is translated into a special type of methionine called formal methionine, or FMET. It's chemically modified. So the abbreviation is FMET. So you guys, if this had been on our lecture exam too, and I said this is E. coli, and I asked you to translate the mRNA, if you put formal methionine, I give you a little bonus point. If you don't, it's fine. It's not a deduction if you forget, but it's just a bonus point if you remember in prokaryotes and bacteria, the first AUG, the start codon, is translated into formal methionine. Any future AUGs are just translated into regular old methionine. Okay? In contrast, you guys, in eukaryotes like human cells, it's simpler for once. The start codon is translated into just regular methionine. So it's just a little quirk of prokaryotes that the start codon will be translated into formal methionine. And again, you guys, don't worry about it. If you remember, it'd be a little bonus point, but not to worry about it. Good. Okay, so let's see here, folks. So again, you guys kind of look in big picture here. In the genetic code, there are 64 codons. 61 are called sense codons, meaning they'll be translated into an amino acid. 3 of them are called nonsense codons. Who do you think the nonsense codons are? The stop, right? There's no amino acid that those codons would be translated into. We're going to see, you guys, the key to this whole event is the transfer RNAs. We'll see how the transfer RNAs are going to link the mRNA codon sequence to amino acid sequence. That's coming up on the next slide. All right, and you guys, here's just the text telling us that in bacteria, the start codon is formal methionine, and eukaryotes is just regular methionine. And this last little point here, you guys, to me, I find this kind of humorous, because when you read the genetic code is degenerate, you know, when I think of something being degenerate, I think of it being of low morals. It's kind of unethical. It's like, how can the genetic code be unethical? Well, in science, degenerate has a different meaning. So when we say the genetic code is degenerate, it means that more than one mRNA codon will be translated into the same amino acid. So what do we mean? So take a look here, you guys. Let's look at the amino acid leucine. LEU is leucine. How many codons will be translated into leucine? 1, 2, 3, 4, 5, 6. 6, right? So that's what we mean by the genetic code is degenerate. More than one mRNA codon will be translated into the same amino acid. And you might say, well, why is this? Well, this is awesome, you guys, because this will provide protection against that low background level of mutations. Remember, folks, when DNA polymerase copies DNA, even though it proofreads, is there still a mistake rate? Yeah, that one wrong nucleotide every maybe 10 to the 9th, 10 to the 10th nucleotides. That's a background mutation rate, and it happens every time our DNA is copied, right? And we know that changes in the DNA sequence will change the mRNA sequence, and what could that change is a consequence? The amino acid sequence of a protein, right? And that could be serious, right? If we introduce a different amino acid, it might mean the protein doesn't fold correctly. So it's thought that by having the degeneracy, this helps protect against harmful effects of that low background mutation rate that's always going on, right? So for an example, you guys, if say in the DNA there was a mutation so that the resulting mRNA codon was changed from CUU to CUC, would that cause a harmful impact on the protein? No, right? Because CUU is translated to leucine, CUC is translated into leucine, right? So that degeneracy is going to protect against that low background mutation rate that's always going on in cells. And again, folks, this was just Yes, let's do this right now, you guys. So I want to give you one of the short answer questions on lecture exam 2, and I want us to practice it, right? So on lecture exam 2, folks, on the short answer section, what I'll do is provide you with a DNA sequence. And I'll just do this randomly, folks. So a DNA sequence, and I'll set it up correctly. I'm not trying to fool you. So I'll set it up with the correct orientation. And let's say the DNA sequence is, and then let me see here. Okay. And then, so this is our short DNA sequence, and then I'll ask you to transcribe the DNA sequence. What does transcribe mean? What does that mean, you guys? Transcribe the DNA. What are you going to make here? RNA, right? And we'll pretend, you guys, it's mRNA. So remember, you have to know that vocabulary. If I say transcribe the DNA, what are you going to make? RNA, good. So you guys help me out. So transcribe the DNA. Okay, remember it's RNA? Good. Good job, you guys. Good. 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 Good job. So, folks, and then I'll say, so transcribe the DNA, and then I'll say translate the transcription product, right? This is getting awkward. So now we're going to translate our transcription product. So what are we going for now? Proteins, amino acid sequence, right? And remember, you guys, you'll have the genetic code table on the exam. You don't have to memorize it. So, folks, help me out here. So, what will the amino acid sequence of our translation product be? Okay, yeah, if it was E. coli, right? If you wanted a little bonus, we would put formal methionine, or guys, you can just put methionine, right? Don't even worry about it. And then GCG. GCG is looking like alanine. And then CCC. CCC proline. Good. And then, folks, if I had a stop codon, are you going to put anything there? You could put stop, or you could put nothing, right? If there was a stop codon here. Okay, so you guys, for sure, that'll be a short answer question on lecture exam 2. All right, so just make sure, if you're having trouble doing this, let us know. Okay, let us help you, because we want you just to nail that question on the exam. All right? Okay. Now, folks, remember how we're peeling the onion? We kind of did the outer layer. We kind of did the most important thing. How do you take an mRNA sequence and translate it into an amino acid sequence? But now we want to start asking, well, how? This is a pretty phenomenal event, right? This cool thing going on in the ribosomes. So we're going to start peeling layers back to find out how is the mRNA base sequence translated into amino acid sequence. And the key, you guys, the key to this is the true translator molecule of the cell. Do you remember what that translator molecule is called? Yeah, T stands for transfer RNA, but is it actually the translator molecule as well? Yeah. So the translator molecule is tRNA. And so it's tRNA, it's single-stranded RNA, it's folded into really complex shape. And just taking a look at the slide, folks, I mean, this is a little bit of the complexity. This is it simplified. This is called a stem and loop structure, right? The stems are stabilized by hydrogen bonds between what? The nitrogenous bases, right? So stem and loop structure. For us, you guys, we're going to make it even more simple because there's 2 sites on tRNA that we're interested in. Okay, so on one end is an amino acid binding site. And the opposite end, you guys, is called the anticodon. So let's take a look. Let's make a simpler cartoon and then take a look at what are these 2 sites doing. So you guys, my cartoon for tRNA is going to look like a fork, like a hay fork or a fork you might use at dinner time, right? So this end is going to be the amino acid binding end. And And I'll just use a circle, you guys, as an amino acid. So I'll just put a circle up here to represent some generic amino acid.
Okay.
So here's my amino acid. And then the opposite part, you guys, this is where it gets totally cool. The opposite part is called the anti-codon. And it's going to have a nitrogenous base sequence. Okay, so here's the anticodon, UAG, and I think I screwed this up, you guys. I wanted this to be, I can't think backwards. I can barely think forwards. Okay, so let me change this anticodon from UAG to UAC. Okay. Now, the function of the anticodon is to bind to complementary codons on mRNA. So, you guys, what is going to be the sequence of the mRNA codon to which the tRNA anticodon will bind? Yeah, AUG. So consequently, we know which amino acid will this tRNA carry. And then the way you solve this, you guys, is you ask, okay, so AUG is translated into which amino acid? And let's say it's not the start codon, guys. Let's say it's just a regular AUG. So let's back up, because remember, you guys, on your exam, you're going to have the genetic code table, right? So if I ask you a tRNA with an anticodon of UAC will carry which amino acid, the trick is you write out the mRNA codon, the complementary mRNA codon, and you look for this mRNA codon on the genetic code table, right? So you guys, AUG is translated into which amino acid? Yeah, right? So boom, you know that's going to be methionine. Doesn't that make sense? Isn't that cool? What a cool system, I tell you. So folks, now what we can do, let's do a couple of possible lecture exam type questions. So see how we went, here's mRNA, there's the amino acid. What is the adapter here? The tRNAs, right? So you guys, let's go in and fill in the tRNAs and do the anticodons. Okay, so folks, the tRNA that is going to bind to the mRNA codon AUG, what's its anticodon going to be? UAC. Good. And which amino acid is it carrying? It was a start before finding. Okay, great. So you guys, what about CGC? What's the anticodon of the tRNA that's going to bind to CGC? What's the anticodon? GCG. So this tRNA is going to carry which amino acid? We look at the mRNA codon and the genetic code table, so we know this tRNA will carry which amino acid? Arginine? Yeah.
All right, now I'm getting thrown off.
Sorry.
Because further we're using the codon to find the amino acid. Now we're connecting what we have as an amino acid to the anticodon.
We're connecting the amino acid to the taxicab. That's the Uber, the Lyft vehicle that's going to bring that amino acid into the ribosome. And furthermore, our Uber Lyft has to know which amino acid to bring based on the mRNA sequence here.
Right. So.
Okay, we'll keep going. Maybe this will help if we keep going. Okay, yeah, no, I know. Okay, we'll keep going, you guys. Let's see what we can do. So, folks, what is the tRNA that will bind to the mRNA codon UUU? Good. And the tRNA that's carrying the anticodon AAA will be bringing in which amino acid? Phenylalanine. How did we know, you guys? We look at the mRNA codon to which the tRNA binds, right? Then we look at the genetic code table. We know that UUU will be translated in phenylalanine. So that has to be the amino acid that tRNA is carrying. Okay. Let me see here. Okay, yeah.
So the codon tells the anticodon what amino acid to go grab.
Okay, and yeah, let me go one step further, okay? And then we'll come back to this, right? Okay, so you guys, so this means that in the cytoplasm, there's all of these tRNAs, all these empty tRNAs, right? So, and let's say they have, okay, let's do anticodons. Okay. So all the different tRNAs, they have to be loaded with the correct amino acid, right? That's the whole key to this event. So it's like, how does that happen? And so you get, whoops. So there's a family of enzymes in the cytoplasm, and the family of enzymes, and this would be a bonus question, folks, remembering the name because it's a mouthful. The family of enzymes that load the correct amino acid onto the correct tRNA, they're called amino-acyl tRNA synthetases. So there would be enzymes that would recognize this tRNA with this anticodon and make sure it gets loaded with which amino acid? Formylmethionine or methionine, right? There'll be another amino-acyl tRNA synthetase that recognizes this tRNA and loads it with which amino acid? Arginine. And likewise, you guys, another amino-acyl tRNA synthetase that recognizes this tRNA with this anticodon and loads it with which amino acid? Phenylalanine, okay? So that's an event happening in the cytoplasm before translation. Does that help a little bit? Okay, yeah, that was a key part. So you guys all right with that, how this is happening? Okay? So let me back up here, you guys, because I want to give you... So first of all, remember, the name amino-acyl tRNA synthetase, that would be a bonus question. So if it's just like, oh, my head hurts, just ignore this one. But I'm trying to remember like what tricky questions I like to ask on the oh, I know, this is the one, you guys, that is really hard. So let's say on the lecture exam, and I usually make this a bonus question to you guys. Let's say on the lecture exam, I say you've got a tRNA that has an anticodon of, let me see here, U, okay? And the bonus question is, which amino acid would this tRNA carry? Now, the common mistake is people look for the anticodon on the genetic code table. Is that what you want to do? No. The trick is, you guys, if that's the anticodon, like I do this even on the key. I pretend, okay, that's my my tRNA, that's my anticodon. And what I really need is to figure out which mRNA codon that anticodon will bind to. And what would it be, you guys? A, right? And then I look for this on the genetic code table. So you guys, the codon AAA will be translated into LYS is lysine. So that would be the answer. And again, folks, that will be a bonus question. So if we had U. What's the amino acid, you guys, this little guy would carry? Lysine, right? Let me make sure. Okay, so it's going to bind to triple A's and triple A's, or yeah, lysine. Okay, and again, that would be a bonus question, but it does kind of help you understand the whole process of translation, knowing how those tRNAs will be loaded with the correct amino acid. They'll bind to the appropriate mRNA codon, bringing the correct amino acid into the ribosome. Now, folks, another little tidbit here is we're peeling back our translation onion. This is all occurring in the ribosome, right? This is all occurring in the ribosome. So remember how we said that the peptide bond will be formed by the ribozyme, right? Is the tRNA bring in the amino acids. The ribozyme is going to form the peptide bonds between the amino acids. Okay. Well, at about the same time that peptide bond is being formed, another cool thing is happening. The link between the amino acid and the tRNA is hydrolyzed, right? tRNA has done its job. It's delivered the correct amino acid. So what's going to happen to this tRNA? It gets kicked out of the ribosome to go get loaded with more amino acids, right? Right? And then again, folks, when we get the peptide bond formed there between our dipeptide and this incoming amino acid, what happens to the covalent bond between the amino acid and the tRNA? It's hydrolyzed, right? Pretty amazing stuff, huh? Okay, so guys, let's keep going. Okay. And so, folks, I'm not going to go over these, but these are like classic lecture exam 2 type questions. So again, the point being, if you're having trouble, you're like, I still don't understand how to do these, make sure you touch base with me, touch base with a colleague, because you guys know your classmates are the best teachers, right? They get where you're coming from. Sometimes as instructors, we don't have the right perspective. But certainly you guys touch base with a colleague or touch base with me or email me, and I'll try to explain it maybe using a different approach, right? Because again, sometimes when we first learn a concept, well, it's like, I don't get it. And then somebody else will explain it a different way. You're like, oh, now I get it. That's not that hard. Okay? Oh, and you guys, just this one, this isn't meant to be tricky. This last one, which anticodons of tRNAs bind to the codons UAA, UAG, and UGA? What are these codons, you guys? They're stop codons. There's no transfer RNA that binds to those codons. Remember, that's a signal for the ribosome to stop translation, disassemble, release the protein, release the mRNA. Translation is over. Yeah. Okay. All right, and then folks, we will, after we do the antibiotics, we will come back to a series of cartoons that actually show the events in the ribosome, right? But first, because I know this gets kind of dry, let's talk about ways we can inhibit 70S ribosomes in bacteria, right? And we know that those substances are going to be helpful as antibiotics, right? So some of this is going to be review. Okay. All right, and big picture first, you guys, there's two types of ribosomes based on size. So do you remember the two types of ribosomes, folks? What are the little ones called? 70S ribosomes, right? What are the big ones called? 80S, good. And folks, we're going to talk about the cytoplasm. Okay, so we're talking about cytoplasmic ribosomes. So who has 70S cytoplasmic ribosomes? Prokaryotes, okay? And remember, folks, prokaryotes would be bacteria and archaea. And since we're focused on bacteria, right? So our E. coli, what kind of cytoplasmic ribosomes will it have? 70S ribosomes. Good, you guys. Now again, if we're talking about cytoplasmic ribosomes, which group of organisms have 80S ribosomes? All eukaryotes, right? Okay, so now, why am I making a big deal about cytoplasmic ribosomes? Where else in your cells would you have ribosomes? Yeah, good, you guys. So in parentheses over here, under 70S ribosomes, right, where else might you find 70S ribosomes? Not in the cytoplasm of these cells, but where? They're structures found in your cells and eukaryotes, so, and you guys said it already. Yeah, mitochondria. So remember, you guys, according to the endosymbiotic theory, mitochondria evolved from what? Yeah, from bacteria. So part of the evidence supporting the endosymbiotic theory is your mitochondria have 70S like ribosomes, just like bacteria. And likewise, chloroplasts have 70S like ribosomes, right? Your mitochondria and chloroplasts, you guys, have circular DNA, just like a bacterial chromosome. So the evidence is really supportive of the endosymbiotic theory. Okay, but if I ask you for cytoplasmic ribosomes, prokaryotes, bacteria, and archaea have what? 70S, and eukaryotes have 80S cytoplasmic. Good job, you guys. All right. Now, for lecture exam 2, folks, I would like you to know the size of the two subunits. So it turns out ribosomes are made up of two subunits, a small subunit and a big subunit. And for lecture exam 2, folks, I just want you to know the subunits for the prokaryotic, the bacterial ribosome. And this makes people crazy, and I apologize. So in the 70S ribosomes of bacteria, the large subunit is called the 50S subunit, and the small subunit is called the 30S subunit. And I know this makes you crazy. When you add them together, right? I know you're going to say 50 and 30 doesn't make 70. And the reason is, you guys, what we're measuring here, this S, is the Svedberg unit. It's a measure of the sedimentation rate. When you spin something at really high speeds in an ultracentrifuge, you're measuring how quickly the substance moves from the top of the tube to the bottom, sedimentation rate. And sedimentation rates are based not only on mass or weight, but they're based on shape, right? So it turns out if you take a prokaryotic ribosome apart and you centrifuge the parts separately, the big one ends up being 50S sedimentation rate, the small one ends up being 30S sedimentation rate. But if you put them together and centrifuge them, you end up with this 70S sedimentation rate. So it's just so you know the math actually isn't wrong. It's just kind of goofy. All right. So, and I think Dr. Svedberg was the scientist that invented the ultracentrifuge, which spins things at incredibly high speeds, and thus the unit was named in his honor. Okay. Now, this is a really messy slide, you guys. So again, just saying prokaryotic ribosome 70S, eukaryotic cytoplasmic ribosomes are 80S. Let me go one or two forward, you guys, to see if I've got a better slide. Okay, this is just showing the two subunits of the prokaryotic or bacterial ribosome. Okay, now this is messy, folks, so let me write it up on the board. This would be a bonus question, but there are two specific ribosomal RNAs that we find in the 70S ribosome, and for bonus, I'd like you to know what their functions are, because to me, I mean, this is really cool stuff. All right, so in the 70S ribosome, remember we have the two subunits, the 30S subunit and the 50S subunit. And each subunit, you guys, is made up of ribosomal RNA and proteins, right? But the really cool thing is in the 30S subunit, there's a special ribosomal RNA called the 16S ribosomal RNA. And its job is to bind to, remember on the mRNA, folks, we said there's a ribosomal binding site. That's the site that the ribosome binds to, and then it's going to start translation as soon as it hits the first AUG. Yeah? It's the 16S ribosomal RNA that recognizes that ribosomal binding site on messenger RNA. It's kind of like the brains of the ribosome, kind of like sigma was the brains of RNA polymerase. So it's the 16S ribosomal RNA that binds to the ribosomal binding site on messenger RNA. So we could argue it's kind of like the brains again. It's telling the ribosome, okay, we're going to bind here, and as soon as we find the first AUG, we're going to start translation. Does that make sense, folks? It functions like the sigma factor on bacterial RNA polymerase, right? Okay, cool. Now, the other thing, it's so exciting. Why do I get excited by these things? Because they're amazing. So in the large subunit, the 50S subunit, again, folks, there's another really special ribosomal RNA, and it is 23S ribosomal RNA, and this is the ribozyme. So you guys, what the heck are we talking about? What's a ribozyme? A catalytic RNA, right? And what's the job of the ribozyme in a ribosome? Formation of the peptide bonds between the amino acids. Isn't that just amazing? So the ribozyme, it catalyzes formation of, that's kind of hard to see, peptide bonds between sugar, the peptide bonds between amino acids during which process? Translation, right? Sometimes we forget, where are we? Isn't that amazing? pretty cool. Okay. And folks, I do apologize because this slide, the way that textbook publisher had organized this and the way I wanted to present it was in a different order. So it's just a bacterial ribosome. The 16S ribosomal RNA of the small subunit is going to bind the ribosomal binding site. And then it's a 23S ribosomal RNA of the large unit that's going to act as a ribozyme and catalyze formation of the peptide bond during translation. Pretty cool stuff. Would those two be bonus questions? Yes. So again, you guys, I know if you're feeling overwhelmed, just don't even think about it, right? All right, and then I think, folks, what we're next going to do is take a look really briefly at actually the process of translation. So before we do, I'd like to take a look at our assembled ribosome. And again, folks, we'll presume this is E. coli. So what's this little red ribbon here? That's the mRNA. And you'll see, you guys, when the ribosome is assembled, there's a groove through which the mRNA is fed through the ribosome. All right. Okay. And then, folks, if we did a longitudinal section through the ribosome, when it's bound to the mRNA, we'll see within the ribosome there are three sites for the attachment of tRNAs. And you guys, on our lecture exam, I'm not going to ask you about these three sites, but I just want to explain it to you so when we look at the cartoons of translation, it makes a little bit more sense. So folks, these three sites within the ribosome, the A, the P, and the E site. The A site stands for the aminoacyl tRNA binding site. The P site is the peptidyl tRNA binding site. And the E stands for exit. This is where those empty tRNAs are going to get what? Booted out of the ribosome. Okay? So folks, let's take a look at then sugar. Okay, you guys, I'm going to skip this and go to the cartoons of the translation, and then we'll come back and look at the antibiotics. I'm sorry, I got ahead of myself there. Apologies. We will come back, I promise you. Okay, so you guys, we're just going to look at a few diagrams of the process of translation. So folks, if this is our mRNA, where initially would the ribosome bind? At the ribosomal binding site, remember? And that's going to be just a little bit upstream of the start codon, right? So I wish they'd extend this a little bit more. But the ribosome scans the mRNA until it finds that ribosomal binding site. It's going to bind there and then start reading the mRNA until it comes to the first AUG, and that's where it'll start translation. Okay. So here we have the ribosome assembled. Here's our mRNA. And to get translation started, folks, this is called initiation. We'll see that the start codon will be in that P site. The next codon will be in the A site. So the ribosome selects the correct tRNA that's going to bind to the AUG. So the anticodon has to bind to the AUG codon. And that tRNA, you guys, is loaded with the correct amino acid. Okay? And then it's going to be over and over and over again. And then the ribosome chooses the tRNA whose anticodon will bind to the next mRNA codon is carrying the correct amino acid. Okay. And then here's a cool event, you guys. The ribozyme, the 23S ribosomal RNA, is going to form what between those two amino acids? Peptide bond, right? And at the same time, the covalent bond linking the amino acid to the tRNA is going to be hydrolyzed. Now, in the old days, they taught us that the ribosome will move one codon downstream, and apparently that's not correct. Apparently the ribosome pulls the mRNA through that groove, right? So in the next step, we can see that the tRNA that's now empty gets moved to the E site. What's going to happen to it? Right? Gets kicked back to the cytoplasm. Now we have our little dipeptide attached to the tRNA in the P site. The ribosome selects another tRNA that will bind to the next codon. That tRNA is carrying the correct amino acid. What's going to form here, you guys? Peptide bond by the ribozyme. What will happen to that bond between the dipeptide and the tRNA? Hydrolyze. Awesome. You got it already. You got it already down, right? There's formation of the peptide bond. Here's that empty tRNA getting kicked out, right? And again, folks, it is almost like the ribosome just moves down the mRNA one codon at a time. choosing the correct tRNA with the correct amino acid, right? So the process we just described, you guys, it just keeps happening over and over and over again, and over and over and over again, until what happens? Yeah, it hits a stop codon. And again, folks, we're simplifying. We're saying when the ribosome hits the stop codon, it will disassemble, releasing the mRNA, and also releasing the polypeptide. So it will finish folding into its functional conformation. Okay? Ta-da, ta-da. We should have music that goes with these. Okay. Okay, folks, so that was kind of our quick little view at translation. So just some little tidbits just to finish up, you guys. Do you recall how we said in prokaryotes and bacteria, the DNA is in the cytoplasm, RNA polymerase is in the cytoplasm, the ribosomes are in the cytoplasm? So you guys remember how we said in bacteria, ribosomes can bind to the mRNA even before transcription is finished and start translating the mRNA even before transcription is finished, right? So this could be called simultaneous or coupled transcription and translation. Can this happen in your cells? No, right? Remember, transcription has to be finished in the nucleus before the mRNA can travel into the cytoplasm where the ribosomes will bind. Okay, so eukaryotic cells, no coupled, no simultaneous transcription and translation, only in prokaryotes. Good. But you folks, what are these little coconuts? Those are ribosomes, right? Okay. So when we have multiple ribosomes translating and mRNA at the same time, it has a fancy name. It's called a polyribosome or a polysome. So you can have polyribosomes formed in both prokaryotes and eukaryotes, right? So in your cells, once that mRNA gets into the cytoplasm, you're going to have multiple ribosomes that will attach. They attach the ribosomal binding site, you guys, and then they start reading. And as soon as that ribosomal binding site is free, another ribosome can attach. And again, it's just a vocabulary word. Polysome, polyribosome, it just increases the rate of protein synthesis. Okay, and now, folks, I think what we're going to do, and I apologize, I should have put these slides in different. We just want to back up and talk about classes of antibiotics that will specifically inhibit the prokaryotic, the bacterial 7S ribosome. And this goes way back, you guys, to our bacterial cell structure lecture. So hopefully it's a little bit of review. So you guys, can you provide the name of four classes of antibiotics that specifically target the 7S ribosome? And you can read the slide, that's totally fine. So tetracyclines, macrolides like erythromycin, azithromycin, chloramphenicol, aminoglycosides. Aminoglycosides, you guys, include gentamicin, kanamycin, streptomycin, right? And just a couple of words of cautions, you guys. Chloramphenicol, usually you try not to use chloramphenicol because in some people it causes aplastic anemia. It can actually shut down bone marrow production of red blood cells. If it's life-threatening, of course, right? But it's probably not the first antibiotic you're going to choose. Aminoglycosides, you guys, you have to be careful because aminoglycosides can be really hard on your patient's kidneys. So if your patient has any kind of kidney disorder, kidney disease, be really careful with the aminoglycosides. And in some people, it can also cause auditory nerve damage, right? So the aminoglycosides, they're not as safe, say, as the beta-lactams. Beta-lactams are really safe unless your patient is allergic to beta-lactams, right? Okay, let me see here. What else, you guys? Now, the next cartoon, don't panic, you guys. You don't have to memorize exactly what each antibiotic is doing. But this I thought was cool, folks, because it's showing us that the different antibiotics have different mechanisms of inhibiting the protein synthesis by the bacteria. Do you need to memorize that? No, you don't, right? But just seeing there's many different ways these antibiotics can interfere with translation in bacteria. And you guys, why is it that at therapeutic, safe levels of these antibiotics, protein synthesis in your cells isn't shut down? Why is that? Because, right, we have the bigger ADS ribosome, so these antibiotics are going to selectively bind to the 70S ribosome, again, at therapeutic levels, right? And again, folks, therapeutic levels, because we don't want those antibiotics to do what in our mitochondria? We don't want those antibiotics to shut down the function of the ribosomes in our mitochondria, right? So we talk about safe levels, therapeutic levels. Good. Okay, we did this. I think you guys, oh, I know, there is one more slide here. Okay, so you guys do remember lecture exam 2 is coming up not next week, but the week after that. And it's a big one. I think it's, I should say this, but I do think it's the most challenging, you guys, because it's going to be metabolism, it's going to be microbial growth, and it's going to be genetics, probably through, what I'm thinking, folks, is the information on lecture exam 2 will stop on Tuesday's lecture, right? Just so, I mean, it's so much information. But the reason I wanted to show this slide is, do you remember back in metabolism how we were talking about enzyme inhibitors? And we had that enzyme inhibitor worksheet. We were talking about penicillin, ampicillin, amoxicillin, the beta-lactams that are irreversible competitive inhibitors of bacterial transpeptidase. And then we talked about clavulanic acid that was an irreversible competitive inhibitor of beta-lactamase. And then we talked about the beta-lactamase resistant beta-lactams like methicillin. Right? Does that sound like distant memories? Right? So I wanted to show you this slide, folks, because I think this slide could be really, really helpful as a study guide before lecture exam 2, because we're going to have so many antibiotics on that lecture exam. So let's just go over it really quick, you guys, as a review. And then we are going to start part 2 of microbial genetics, folks, today. Okay, so folks, do you remember in our microbial genetics section, we said when E. coli or bacterium is ready to split in 2, they have to copy their chromosome, right? And we said normally the chromosome is in which kind of state within the cell? Supercoiled, right? So we said the cell has to relieve the supercoiling, and which bacterial enzyme relieves supercoiling of the chromosome? Bacterial gyrase, right? So do you remember an antibiotic that will inhibit bacterial gyrase? Ciprofloxacin was a specific antibiotic. It's an example of a quinolone or fluoroquinolone. Good, you guys. So we're going to shut down DNA replication. That will stop the bacteria from replicating. Now, do you remember another antibiotic that will inhibit bacterial RNA polymerase? It will shut down bacterial transcription. It's the one that turns your urine, your feces, your sweat, your tears, your saliva red, orange. Good, rifampin or rifamycin, good. So that's going to shut down bacterial transcription, so the bacterium can't make any proteins now, right? Good. Okay, so that was transcription. And then, folks, do you remember, can you give 4 classes of antibiotics that will inhibit the bacterial 70S ribosome? Tetracycline, the macrolides like erythromycin, azithromycin, okay, chlorphenicol, aminoglycosides, good. And which process will which process would those antibiotics inhibit? Translation, good. Good job, you guys. And now this is one we might have forgotten, folks. Do you remember, how did we describe beta-lactam antibiotics? Irreversible, competitive inhibitors of bacterial transpeptidase. Does that ring a distant bell? Right. And then, folks, we said there's 2 more competitive inhibitors of bacterial enzymes involved in folic acid synthesis. Do you remember those sequential inhibitors? Good. Sulfa drugs like sulfamethoxazole and trimethoprim. Awesome. Shuts down folic acid synthesis. Folic acid is required for DNA and RNA synthesis. Good job, you guys. Excellent. Okay, so I just thought that slide, again, you guys, I love these concept maps. I love bringing a lot of information together. I thought that slide could be really, really helpful preparing for lecture exam 2. Okay, and I think that's the last slide, folks, and microbial genetics, part 1. So folks, and I know everyone is so tired. We're officially halfway through, right? We're going to do this. And so we're going to continue microbial genetics. And again, folks, I know this is like the unit. It never ends, right? Ooh, what row? Okay, you know what? I think Oh, here we go. Okay, sorry. I thought maybe I hadn't downloaded it. Okay. All right, folks. So this is going to be genetics, microbial genetics part 2. And I just realized I forgot a toy. That's all right. Okay, I can fake a toy up here. All right. So you guys, apologies. Let me double check, because I think I did some modifications on the microbial genetics I posted on Canvas. So you guys, I apologize. I should have done this ahead of time, and I didn't. But let me make sure I'm getting the right copy. No way. No way, no way, no way. You silly. Don't do that to me. It's all right. We can use the older one, but I think I had. Okay, you guys, we'll let it be. We'll let it be that way. You guys, just be aware that I think on the microbial genetics part 2 that I posted on Canvas, I did eliminate a little bit of stuff. Like, for example, on this first slide, I think I might have eliminated this. I could be wrong. Okay. Okay, so folks, the topics we're gonna tackle in microbial genetics part 2. Again, we're gonna remain with bacteria as our little model organism. We're gonna talk about control of gene expression. That is, how do bacteria turn off and turn on transcription of their genes, right? And then we're gonna look at, very briefly, mutations, right? And then the last topic will be horizontal gene transfer. Okay, so this isn't quite as detailed as like DNA replication and transcription and translation, but we're going to see these are really cool topics in us understanding how bacterial populations survive, how they adapt, how, for example, overuse of antibiotics, how we are encouraging the spread of antibiotic resistance genes. So these have some really cool practical applications. Okay, so we're just gonna go forward, GAs. So again, always in the back of our mind, we always have the roadmap of the central dogma of information flow in cells. And what we want to remember, folks, is as biologists, we presume that when organisms are growing out there in the world, it's always a bit of a struggle. It's a struggle for resources. It's a struggle for nutrients, a struggle for living space, right? And so as biologists, our understanding is that organisms that are wasteful, right? They're wasteful of their nutrients. They're wasteful of their building blocks. They will have a lower survival rate than neighboring microbes that aren't wasteful, that are conservative, meaning they're not going to waste energy sources or building resources, right? So there's this pressure for organisms not to be wasteful. And this becomes important then, folks, because we've just talked about transcription and translation, gene expression, where information in DNA eventually will guide protein synthesis. So the question now, you guys, we want to ask ourselves is, what if a bacterium doesn't need some protein product? What if it doesn't need a specific transport protein? What if it doesn't need a specific enzyme? Is it beneficial for the microbe to continue transcription and translation of a gene for a product it doesn't need, or would that be considered wasteful? It's wasteful, right? And what if, in contrast, we have a bacterium that can turn on and turn off transcription based on the need for the protein product? Would that microbe have an advantage over its wasteful neighbor? Yeah, it would. It would have a survival advantage, right? So you guys, these cool strategies bacteria use to turn on and turn off transcription, they've evolved because it gave the bacteria survival advantage over their more wasteful neighbors. So we want to take a look at the first way. This was the first time that a strategy the bacteria used to control gene expression was discovered. And this was by two really famous French microbiologists, Jacob and Monod. And we're going to use their model. They described how E. coli can turn on and turn off genes that are involved in lactose transport and lactose metabolism. Before we get there, though, we have vocabulary, right? We always have to have vocabulary. So you guys, in an E. coli, we know there's some genes that encode proteins that the bacterium needs all the time. For example, the enzymes involved in glycolysis, the bacteria needs to make those enzymes all the time, right? It always has to be carrying out glycolysis. So those genes are constantly transcribed and translated. We would say that there is constitutive expression of those genes because the gene products, those proteins, are constantly needed, right? So those genes are constantly being expressed, translated and translated. Now, in contrast, you guys, there's some genes whose products, whose proteins, are only needed under special circumstances. And usually these are genes for transport proteins or genes for catabolic enzymes, enzymes that are going to take a big molecule and tear it down into smaller parts. So you guys, let's set the scene. Let's say you're an E. coli, and we're going to use lactose here. And let's say you're growing in some environment, and there's no milk in your environment. There's no lactose. Does it make survival sense to transcribe and translate the genes for lactose transport proteins and for beta-galactosidase, the bacterial equivalent of lactase? If there's no milk, no lactose in your environment, does it make sense to spend all that energy, all those building blocks to make lactose transport protein and bacterial lactase and no lactose? No, right? So it turns out, you guys, the genes for lactose transport proteins and the gene for beta-galactosidase, that's the bacterial lactase, they are what we call inducible. They will only be synthesized when a signal molecule is present. What would be a good signal molecule? Lactose, right? So, inducible genes are often genes for transport of a specific substance, or they're catabolic enzymes that are going to help break down a specific substance, right? Inducible, they get turned on when a signal molecule is present. So that's what we're going to focus on, you guys, because that was the first method that was discovered by which bacteria, E. coli, can turn on and turn off gene expression through transcription. So you guys, there is a third category of genes. We are not going to talk about them. They're called repressible genes, and we are not going to talk about them, right? And it's not on exam 3. It's going to be not on exam 2. So we're only going to, I would only ask you about constitutive genes and inducible genes. Okay. So folks, to talk about inducible genes in E. coli, we're going to be talking about what's called the Lac Operon. So this is a brand new term. So let's start anew here. And I forgot my Lac Operon of science in my office, you guys, but we'll just do it on the board, and then next Tuesday I'll bring it in. And it's a hands-on kind of a kinesthetic model, which I think is really helpful for a lot of us. Okay. Okay, so again, folks, we're starting Microbial Genetics, Part 2. This is the second PowerPoint, okay? And we're going to talk about control of gene expression. And remember, you guys, we said gene expression, 2 stages, transcription and translation. So we're going to focus on control of transcription. And just as a reminder, folks, in transcription, what's the template? Good. DNA, right? And what are we going to make? RNA, and let's say specifically, you guys, mRNA, right? And what is this process called? Transcription. In bacteria, which enzyme perform transcription? RNA polymerase, awesome. Good job, you guys. Okay, just so we can kind of review the basics, okay? So folks, again, this was the first example of control of gene expression discovered in E. coli by Jacob and Monod. And they describe control of gene expression in what's called the Lac Operon of E. coli. So there's a couple things we need to describe here. What do you think Lac is an abbreviation for? Lactose, right? And lactose is what, you guys? Yeah, it's a disaccharide found in milk. Mammalian milk, right? A disaccharide, what are the two components? What are the two? Glucose, right? Covalently linked to galactose. And what's that bond called? A glycosidic bond, right? If we're going to digest lactose, you guys, must that glycosidic bond be broken? Yes, it must be hydrolyzed. In humans, what's the name of the enzyme that will break the glycosidic bond? Lactase. What's the fancy term in E. coli? Yeah, beta-galactosidase. Okay, just again, beta-galactosidase is the bacterial equivalent of human lactase. Okay, just the background information, you guys. Good, okay. Lactose, little review. But operon, you guys, this is a brand new term, okay? So operons are genes which can be turned on and off. Okay, so let's make a real simple operon here, you guys. Now, you might want to start with a brand new piece of paper, landscape or horizontally, right? Because we're going to make this big operon. So this is at the level of DNA, you guys. It's always confusing. Are we talking RNA or DNA? Well, now we're back at the chromosome, right? So this is single-strand DNA template of the chromosome. And I'm going to make it kind of long, folks. Right? Okay. And to make my Lac Operon, okay, well, first of all, let me put in the structural genes. When I say structural genes, these are genes that encode information for proteins. And in the Lac Operon, there's 3 structural genes. And they're named Lac Z, Lac Y, and Lac A. So let's put those in here first, okay? So here's the Lac Z gene, the Lac Y gene, and the Lac A gene. And we'd like to know, well, what proteins will be made when these genes are transcribed and translated. So the Laxity Gene, you guys, when it's transcribed and translated, this is going to be the beta-galactosidase. What does that do again? Yeah, it's the bacterial equivalent of lactase. It's going to break the glycosidic bond in lactose, so the two monosaccharides, the 2 sugars, can be fed into glycolysis. Good. The Lac-Y gene, you guys, this is a lactose transport protein. Right, to bring lactose into the cell. And the Lac-A gene, you guys, this is called a transacetylase, and we're going to ignore it. To keep our story simple, you guys, we're going to focus primarily on the Lac Z, beta-galactosidase, and Lac Y, the lactose transport protein. Okay? All right. So now, what's cool, you guys, if this is the DNA, there is a transcription terminator just at the end of the 3rd structural gene. So you guys, if RNA polymerase binds and transcribes these 3 genes, it will all end up on one mRNA, and thus that mRNA is called polycistronic, right? So mRNA that carries information for 2 or more proteins, polycistronic, right? Okay, just so we have that under our belts. Now, folks, remember in transcription, right? What's the enzyme again, folks? What's the enzyme that carries out transcription? Okay, so the enzyme that carries out transcription is RNA polymerase, right? And do you remember, you guys, in RNA polymerase in bacteria, it has 2 parts, the sigma and the core, right? What's sigma for? Why do we call sigma the brains of RNA polymerase? What is it going to bind to? We said it was kind of like the start signal in transcription. What was that special DNA sequence called? The promoter. Good. So guys, let's put a promoter here. So far you're like, okay, this all seems pretty familiar, right? But to make this an operon, you guys, we have to put a new DNA sequence between the promoter and the structural genes. And this new DNA sequence, it has a fancy name. It's called the operator. And we could argue, you guys, the operator is the on-off switch for transcription. Okay, now let's see how this works. Now it gets a little complicated. Let me just see my next picture, you guys, here really quick, sorry. Okay, I'll back up here. All right, so what we've written up here, the promoter operator Laxity Y and A gene. This is the Lac Operon. Okay, structural genes, promoter, and an operator. Now, on the bacterial chromosome, outside of the Lac Operon, is another gene. And this gene is called the Lac gene. It too has a promoter. So you guys, are you all right if I put P equals promoter? Is that okay? So the Lac gene, it has a promoter, right? Here, right? And what's interesting is the Lac gene is expressed constitutively. What does constitutive mean? Constantly transcribed, constantly translated, right? So this is constitutive expression, meaning it's constantly being transcribed and translated. So that means, you guys, there's constantly lots of Lac repressor protein in the cytoplasm. It's constantly being made. Now, the Lac repressor protein, you guys, it's kind of cool. It has 2 binding sites. Okay, it has 2 binding sites. Let me see how they've cartooned it here. That's an okay binding site. Okay, so you guys, we'll say that the Lac repressor protein, let's say it has a shape like this. And I'll put a big I for the Lac repressor protein there. And you guys, it has one site, it has a DNA binding site where it can bind, it binds the Lac operator sequence. And then it has another site down here, you guys, and this is going to be the inducer binding site. And the inducer is going to be, we'll call it lactose allolactose. That's going to be our so-called inducer. Okay, so you guys, let's set the scene here. So let's say our E. coli is growing in the absence of lactose. Okay, so let's say our E. coli is growing in the absence of lactose. So there's no lactose in the environment, you guys. Now remember, we're thinking as evolutionary biologists. Would it make survival sense for E. coli to transcribe and translate all these genes when there's no lactose in the environment? Does that make survival sense? No, it's wasteful, right? So when there's no lactose in the environment, the Lac repressor protein comes over here and it binds to the operator. I'll make it great big, you guys. It binds to the operator. So this is we'll put the Lac repressor. What does repress mean? So you stop, to hold down, prevent, like an inhibitor, right? So you guys, if we have this big protein stuck to the operator, and then our RNA polymerase of science binds to the promoter, is that literally a roadblock there? Yeah, it is, right? So you guys, in the absence of lactose, will the Lac Operon genes be expressed, transcribed, and translated? No. Is that good for survival? Yeah, right? Why waste the resources, the building blocks, the ATP, making genes you don't need, right? Okay, so you guys, in the absence of lactose, what will the Lac repressor protein do? It's going to bind to the operator, literally make a roadblock, so RNA polymerase can't get past it to transcribe the Laxity Y and A genes, right? We turn off transcription. Does that make sense? No lactose, no transcription of the Lac Operon genes. So far, so good. Okay. But you guys, let's say now we have a change of environment. Let's say the E. coli, I don't know, maybe it's living in your intestine. You normally don't eat dairy products, right? And suddenly you just can't stand it. You have a big bowl of ice cream, right? All right. So let's say there's a change of environment. And now there is lactose present. So you guys, lactose is a wonderful source of energy and carbon. So if you were E. coli, do you want to transport it into the cell and break it down and use it as a source of carbon and energy? Would you like to do that? Yeah? Yeah? But you got a problem, right? How can you get the LAC-Z and Y genes expressed, right, if this repressor is in the way? So what do you want to do, you guys? Do you want to somehow get rid of that LAC-I repressor protein, get it to fall off? Okay? And there's a cool way to do this. So you guys, the lactose, a little bit is transported into the cell. A little bit, a little tiny bit, a wee bit. No, wee's not very scientific. A little lactose transported into the cell. And within the cell, and you guys, this level of detail you don't need to know, but just to make it official. So within the cell, the lactose gets converted, just a minor chemical modification, into this substance called allolactose. And folks, for our lecture exam 2, if I ask you what is the inducer of the Lac Operon, that is, which molecule turns on transcription, you can tell me either lactose or allolactose. These are the inducers of the Lac Operon. And what I mean by that is that they're going to turn on transcription. Now, how can they turn on transcription if the LAC-I repressor is blocking RNA polymerase's way? What do you want to do? You want to get that LAC repressor off, right? And the way this happens, we'll use a little triangle here, you guys, for the allolactose, right? It's going to bind to what? Yeah, it binds to the allolactose. And again, these are called the inducers. They bind to the repressor protein and get it to change shape. Kind of like those allosteric enzymes. The repressor changes shape, and what happens? It falls off, right? And again, you guys, I apologize here. Here's the lactose, allolactose. The repressor, and I'll make it really dramatic, you guys. Let's say we just totally wipe out the DNA binding site. So here's our inactivated repressor. Here's the lactose, allolactose, binding to the repressor. It's changed shape. This is a LAC-I repressor. It's changed shape, and what's happened as a result? It falls off, right? So you guys, have we removed the roadblock for RNA polymerase now? If this thing has fallen off, right? We got rid of the roadblock. And so now you guys can, forgot my toys, now can RNA polymerase bind to the promoter? Is there a roadblock there anymore? Nope. So it binds to the promoter, and now it can transcribe the LAC-Z, Y, and A gene into that polycistronic messenger RNA. The ribosomes will bind and translate the mRNA into lactose transport protein and beta-galactosidase. Okay? So you guys, is this Lac Operon in the presence or absence of lactose? No lactose, right? Repressor protein. And then you guys, here we see the allolactose, green, good, binds to the repressor, gets it to change shape, repressor falls off, and now RNA polymerase can transcribe those genes. Okay? And folks, I know that this is maybe not that exciting, but we're going to see that in nature there are inducible toxin genes in bacteria. There are inducible antibiotic resistance genes in bacteria. So when we come back on Tuesday, we'll make some practical applications of this model, okay? All right, guys, thank you so much. And I'll see half of you at 1, and the rest of you, I hope you have a really, really good weekend, right? Really good weekend. Oops, had to stop this.
Was there a third handout?
No.
Just
No.
these two?
Yes.